that can turn on that gene? Well, initially this activator protein is in an inactive state. And so it's not down there in the nucleus turning on the gene, expressing the gene. But when he eats the carbohydrate-rich meal, one of those carbohydrates is going to be detected on this stomach cell by this receptor right here. And so now the stomach cell is aware that Rod is eating this carbohydrate-rich meal, and it starts a signal transduction pathway. The first relay protein gets activated by the receptor, and it activates the next relay protein, which activates the next relay protein, usually by kinasing, by adding phosphates. And eventually, the activator protein gets activated, so to speak, and now it can go down into the nucleus and bind the control elements of the gene. And so now this gene is going to be expressed, and we're going to get the carbohydrate digesting enzyme made. Oops, go back. Alrighty. So for environmental specific expression and for developmental specific expression, there are essentially molecular signals that tell the activator proteins to become activated. Okay. Questions so far? Clear as mud? Okay. Well, as I've presented this scenario so far about differential gene expression, I've been emphasizing the presence of the activator proteins. And I've said if the activator proteins are present, then they bind to the control elements. And then that gives you expression because the transcription factors are always already there. And so it comes down, as I've shown you so far, it comes down to are the activator proteins present or not. If they are, you get gene expression, and if they're not, you don't. For some genes, it's exactly like that. It just comes down to are the activators proteins present or not. For other genes, there's yet another protein that can be involved. For other genes, there are repressor proteins that get involved in the transcription expression. And these repressor proteins, as you might guess from their name, decrease expression of the gene. The activator proteins, if they were there, they increased expression. These proteins are there to decrease expression of the gene. They're sort of the evil twins of the activator proteins. And we ran into repressor proteins before, or at least the concept of them, when we talked about bacteria and their operons. You might remember like the lac operon and the tryptophan operon had a repressor protein that could block RNA polymerase from binding to the operator of the operon, essentially the promoter of the operon. Oh, well, I gave away the question I was going to ask. I was going to say, in bacteria, where do the repressor proteins bind? And you were supposed to say, to the promoter, Mr. Reedens, or the operator, which is the name of the promoter for an operon. But in eukaryotes, it's a little different. The repressor proteins don't bind to the promoter, the sequence of the promoter. The repressor proteins can bind to the control elements in the enhancer region. And so what the repressor protein can do is simply block off those control elements so the activator proteins can't get at them. Here's one of these repressor proteins. It just binds to those same sequences, the control element sequences, so the activator proteins can't get at it, and hence the repressor protein decreases transcription of the gene. Pretty straightforward. That's the way a lot of these repressor proteins work in eukaryotes. There are other types of repressor proteins that also lead to a decrease in transcription of the gene, but these other types of repressor proteins don't bind to the control elements. These other types of repressor proteins change the region of the chromosome around the gene so that the activator proteins and the transcription factors just can't get in there and bind. And let me explain what I mean by that. Earlier in the semester, I showed you a picture like this. This is supposed to be a cell, and there's the nucleus, and inside the nucleus are the chromosomes. And you can see the chromosomes as linear pieces of DNA. But you might remember you only really see them as linear pieces of DNA like this when they are condensed, which means wrapped tightly around the histone proteins. And remember, you only see them like that during prophase, metaphase, anaphase, and telophase, when the cell is actually going through the nuclear division. Most of the time, the cell is not in prophase, metaphase, anaphase, or telophase. It's in interphase. Matter of fact, many cells in your body are stuck in interphase, in GO phase of interphase. And so most of the time, you can't see the chromosomes because they're not wound tightly on the histones like this. Most of the time, the DNA just kind of spools off the histones and spreads out throughout the nucleus. It's all diffused and spread out. And we call that chromatin. Chromatin is the DNA that's become mostly off the chromosomes. And this is good because when it's all spread out like that, the DNA is very well spread out and open, so the transcription factors and the activator proteins can get to the genes very easily. Well, what they found is when they look really closely at the chromatin, some of it is spread out. Some of the chromatin is indeed spread out and not really bound up on the histones, but other regions of the chromatin is spooled tightly around the histones. Here's a close-up microscopic image of a real cell's chromosome, and here you can see a bit of the DNA there. So parts of it are sticking out and all loose and just sort of flapping around in the liquid inside the nucleus, but other parts look more tightly condensed, so those are parts that are still wrapped up around the histones. And the whole thing is still considered chromatin, but they give different names to the two regions. The part that's still loose and not very much well on the histones, not on the histones really, is called the euchromatin. And the part that still remains pretty tightly bound up on the histone proteins, is still tightly wound up, so to speak, is called the heterochromatin. And as you might guess, you have very different levels of transcription in these two regions. In the euchromatin, imagine it looking like this. The purple little balls there are the histone proteins, but a lot of the DNA is not spooled around them. It's free and open like this, and so the transcription factors and the activator proteins and the RNA polymerase II can get in there very easily and express those genes very efficiently. In the heterochromatin region, it's just the opposite. The DNA is almost entirely spooled up around the histones, so there's just no space in there for the transcription factors and RNA polymerase II and the activator proteins to get in, so you get very low levels of gene expression. And so what they found is, to relate this back to differential gene expression, that the cells can modify the histones in the area of a gene to either open up that DNA, change it, that is to change it into euchromatin, so those genes are expressed more, or if the cell decides it doesn't want to express a gene, it modifies the histones in the region of that gene to turn them into heterochromatin. And so you get less expression of that gene. You're essentially modifying the chromatin as a way of regulating specific genes in the genome. And there you see it. So the genes in this region are not going to be expressed well, and the genes in this region are going to be expressed well. And the cell can move between the two states around any particular gene. Now, here in your notes, I just said that there are these enzymes that modify the histones to change the DNA between euchromatin and heterochromatin to regulate gene expression. You don't have to know exactly how the histones are changed, but if you're curious, it's kind of a interesting way. There's a functional group called an acetyl group that can be added to the histones or removed from the histones. So you call these acetylated histones. and the acetyl group has a negative ionic charge on it. And remember that the backbone of DNA is negatively charged also. And since like charges repel each other, as long as there are acetyl groups on the histones, they push away the DNA, and that tends to spread the DNA out, and you get euchromatin. But when the cell removes the acetyl groups, now the histone proteins are no longer negatively charged, and so they don't repel the DNA, and it's able to wind up tightly on the histones like that, and you get heterochromatin. You buy it? Alrighty. Alright, so these repressor proteins, some repressor proteins just work by binding to the control elements and blocking the activator proteins, but others actually change the chromatin from euchromatin to heterochromatin to decrease expression of a gene. There's yet another trick that cells have to do differential gene expression, and it involves actually changing the DNA of the gene that they're trying to regulate a little bit. It's called methylation of DNA. DNA is G's and A's and C's and T's. You know that. And this is what the nitrogenous base of a C, a cytosine, looks like. This is normal cytosine as you see it in most of your DNA. If a cell decides it doesn't want to express a certain gene, it can send in enzymes to that particular gene and do this to some of the bases. It's called methylating the DNA, because this group that's been added right here, the CH3, is called a methyl group in organic chemistry. And by adding that methyl group, it changes the structure of the gene such that the transcription factors and the activator proteins no longer want to bind to it. So it's a way of turning off a gene. As long as the methyl groups, the DNA is methylated, the gene is not going to be expressed very well. And so there's kind of a cartoon of that. As long as those methyl groups are stuck on there, that particular gene will be inactivated. And if the methyl groups are removed, then the gene is now in an active state and can be expressed. So this is I ripped off this figure from the internet somewhere, but it illustrates two ways that by essentially tweaking with the DNA that the cell can regulate the gene expression. One is by methylating the nucleotides, and the other is by modifying the histones to wrap the DNA either tightly or loosely around the histones to regulate gene expression. The ability of the cells to regulate gene expression by methylation and by modifying the histones are fairly recent discoveries within the field of biology. These really weren't known or understood even just a few decades ago. And they're still sorting out exactly how all this works. But in your readings, you may come across a term, which is this, called epigenetics. The prefix epi means above or beyond. And so what they're saying here, the reason why they coined this term is Mendelian genetics, as we discussed them so far, aren't always sufficient to understand gene expression. And what I'm saying by that is, let's say we were talking Mendel's pea plants. If this is a capital P, you would expect both of these to be purple plants, remember, because they have the dominant allele. But what this field of epigenetics is saying is that if these dominant alleles become methylated, that means they're no longer going to be expressed very well, so they start acting like recessive alleles. They start acting like little p's. And so you can't use standard Mendelian genetics, because you would think this would be purple and this would be purple, but they wouldn't be, because the DNA has been modified. So I guess in a sense it's a type of non-Mendelian genetics. The phenotype you get, like purple or white flowers, is not just dictated by what genotype you have, homozygous dominant, homozygous recessive. You also have to look at the structure of the DNA. And that's why they call it epigenetics. And as I said, they still don't fully understand all the nuances of how epigenetics works, but the methylation pattern they're discovering can be passed on from one generation to the next. And so it almost is as if the gene is being converted from a dominant to a recessive allele. They've done some studies where if one generation, let's say, goes through a period of starvation to help their bodies deal with the starvation conditions, certain genes become methylated, I guess, somehow that are involved in metabolizing the nutrients. And then the offspring of the people who went through that starvation, the next generation, the F1 generation, so to speak, retains that same pattern of methylation, even a generation or two later. So anyway, it's got the modifications of the DNA, the methylation histone patterns, can be inherited. So it is a form of genetics. All right, well, let's see. So up to this point in this lecture on genomes, I've been hammering away really at transcriptional level gene regulation.